Welcome to the Deep Dive, your shortcut to understanding the complex world around us. Today, we're tackling something truly amazing, just astonishing when you think about it.
Yeah.
How did this incredibly complex being with trillions of specialized cells, start out as just one single fertilized egg?
It really is a biological marvel that one tiny cell somehow contains all the information, all the programming to divide, to specialize into muscles, nerves, skin, everything,
Yeah.
and then arrange all of that perfectly.
Exactly, and that's what this deep dive is all about, the incredible precision of gene regulation that makes it all happen. We'll explore how cells get their specific jobs, how organisms get their shape, the power of stem cells.
And also what happens when that control system breaks down, leading to diseases like cancer. It's fundamental stuff.
Think of this as your guide to the basic blueprints of life, from, a fruit fly all the way to us. We're drawing from a key section in a major biology textbook, breaking down the concepts so you can follow along easily. No pictures needed.
Right, we'll paint the picture with words.
Okay, let's start right at the beginning. That single fertilized egg, the zygote, transforming into an embryo, then an adult. It's not just cells dividing like crazy, is it? The source material really hammers home that it's a tightly regulated program.
Absolutely. It's a program of gene expression. Genes turning on, genes turning off, in exactly the right sequence at exactly the right time. It involves pretty much every level of gene control we know about.
And it all boils down to three core processes that are all happening together, right?
That's right. First, you've got cell division, just making more cells, the initial multiplication.
But like you said, that alone would just make a blob. So step two.
Cell differentiation. This is where the magic happens, where cells become specialized. A liver cell becomes a liver cell, a neuron becomes a neuron. They take on specific structures, specific functions.
And the wild part here is that, generally speaking, almost all your cells have the exact same DNA, the same genetic instruction manual.
Precisely. So it's not about having different genes, it's about using them differently. That's differential gene expression. Which genes are active, which are silent. It's like musicians in an orchestra all have the same sheet music, but the violin plays its part, the trumpet plays its part.
Great analogy. Okay, so division, differentiation. What's the third piece?
Morphogenesis. This is literally the creation of form. It's how those specialized cells get organized in 3 dimensions, into tissues, organs, the whole body structure. It's the physical shaping process.
So how do those very first cells know what to do? Where do the initial instructions come from? Our source mentions 2 key things. First up, cytoplasmic determinants.
Right. Think of the unfertilized egg. Its cytoplasm, the stuff inside, isn't uniform. The mother packs it with specific messenger RNAs, proteins, other molecules, but distributes them unevenly.
Ah, so they're already positioned like little instruction packets placed in specific spots.
Exactly. So when the zygote starts dividing, the daughter cells inherit different combinations of these determinants depending on where they form in that original egg cytoplasm. That initial molecular package helps determine their fate, guides their early gene expression. It's a blueprint laid down by the mother.
Wow, okay, so that's the preloaded information. What's the other source?
Inductive signals. As the embryo grows and you have more cells, they start talking to each other. Seriously, they communicate. This can be through direct contact, molecules on their surfaces, or by sending out chemical signals like growth factors that nearby cells pick up.
And these signals, they induce changes.
Yes, they induce changes in gene expression in the receiving cells. One group of cells can essentially tell another group, okay, you guys need to become nerve tissue, or you're going to form part of the lens. It's dynamic guidance.
So you have the initial setup from the determinants and then ongoing communication shaping things. This leads to an interesting point, the difference between determination and differentiation.
Right. Determination is the commitment. It's when an embryonic cell locks onto a specific fate, like deciding, I will become a muscle cell. Importantly, this happens before you can actually see any change in the cell. It's a molecular decision, an internal commitment.
Okay, so it's decided its future, and then differentiation is?
Differentiation is the process of becoming that cell type. It's when the cell actually starts making the specific proteins that define its function and structure. Liver cells start making albumin, lens cells make crystalline, muscle cells start pumping out actin and myosin. Now you can actually see the specialization.
Let's make that concrete.
Yeah.
The book uses muscle cell development as a great example. You start with these embryonic precursors.
Myoblasts. Well, before they're myoblasts, they're precursor cells that could potentially become several things: muscle, fat, cartilage.
Right. But then something pushes them towards muscle fate. They become committed myoblasts. How does that happen?
It involves these crucial genes called master regulatory genes. A key one for muscle is called myoD. The protein made from this gene, the myoD protein, is a transcription factor.
Meaning it controls other genes.
Exactly. It binds to specific DNA sequences called enhancers near the genes needed for muscle development and turns them on. It's like a master switch that activates the entire muscle building program.
And isn't there a clever feedback loop, too?
Yes. myoD actually activates its own gene as well, so it reinforces the decision, locks the cell into the muscle fate. It's a positive feedback loop. Eventually, these committed myoblasts fuse together to form the long, multinucleated muscle fibers that do the actual contracting. It's a beautiful example of sequential gene regulation.
Okay, so cells are dividing, specializing. But development isn't just about growth. Some cells are actually programmed to die, right?
Absolutely. It sounds a bit morbid, but apoptosis, or programmed cell death, is incredibly important. The word literally means falling off, like leaves from a tree in autumn.
And it's not messy like a cell just exploding.
Not at all. It's very orderly. The cell's DNA gets chopped up neatly, organelles break down, the cell membrane bulges out in little sacs, that's called blebbing, and then scavenger cells come along and basically vacuum up the fragments. No inflammation, no damage to neighbors. Very tidy.
Why is this necessary?
For so many reasons. During development, it's crucial for shaping things. For example, the space between your fingers and toes, that was originally webbed tissue that was removed by apoptosis. It's also vital for normal nervous system development. And in adults, it gets rid of cells that are infected, damaged, or just old.
And this isn't a new invention in evolution.
Nope. The basic machinery, the genes involved are ancient. You find similar apoptosis genes in tiny worms like C. elegans, where scientists first mapped out the precise lineage and programmed death of every single cell, all the way up to us mammals. It's a fundamental process.
Okay, so we've got individual cells sorted, but how does the whole organism get its overall structure? Like, how does a head end up at one end and a tail at the other? How do limbs form in the right place? That's pattern formation, isn't it?
Exactly. Pattern formation is all about setting up the body plan, the overall 3D arrangement. And it relies on cells getting positional information.
Like a cellular GPS.
Kind of. Molecular cues tell a cell where it is relative to the main body axis, head to tail, anterior, posterior, back to front, dorsal, ventral, left to right, and also relative to its neighbors. And these cues, they come from those cytoplasmic determinants and inductive signals we talked about earlier.
The classic example here is the fruit fly, Drosophila. Why are they so important for understanding this?
Well, they're easy to breed in the lab, short generation time, and their bodies have this clear, segmented structure: head, thorax, abdomen. And crucially, researchers discovered that the basic body axes in a fly are set up before the egg is even fertilized, thanks to those maternal cytoplasmic determinants.
And this led to some really groundbreaking genetics. Edward B. Lewis.
Yes, a pioneer. Starting way back in the 40s, he studied these bizarre mutant flies. Flies with legs growing out of their heads instead of antennae, or flies with an extra pair of wings.
Wild, like biological mix-ups.
Exactly. Lewis identified the genes responsible, calling them homeotic genes. These are master control genes that specify the identity of body segments. His work was the first real proof that specific genes orchestrate the development of body parts in their correct locations. A huge leap.
And then later, Nuslin, Volhard, and Vicios built on that.
They did. In the 80s, they undertook this massive systematic screen to find basically all the genes that affect segmentation in the fly embryo. It was incredibly ambitious. Think about it, thousands of genes, and many mutations would just kill the embryo, making them hard to study.
How did they manage that?
Through incredible dedication and clever genetic screening techniques, they identified hundreds of key genes involved in setting up the fly's body plan. Their work, combined with Lewis's, gave us this incredibly detailed molecular map of early development, and they all shared the Nobel Prize for it in '95.
And their work really highlighted maternal effect genes, right? Also called egg polarity genes.
Yes, these are genes from the mother whose mRNA or protein products are deposited into the egg as it forms in her ovary. So the mother's genes set up the initial coordinates for the offspring's development, regardless of the offspring's own genes for those particular traits.
The famous example being bicoid. What happens if the mother has a faulty bicoid gene?
Bicoid means two-tailed, and that's exactly what happens. The embryo fails to develop a head and anterior structures. Instead, it develops posterior structures, basically, a tail at both ends.
Two tails, no head. Bizarre.
It led to a key idea, the morphogen gradient hypothesis. The idea is that the bicoid protein itself acts as a morphogen. It's a substance that diffuses through the early embryo, creating a concentration gradient.
So high concentration at one end, low at the other.
Exactly. They found the bicoid mRNA is parked at the anterior head end of the egg by the mother. After fertilization, the protein is made and it spreads out, forming a gradient highest concentration at the future head, lowest at the future tail. The concentration of bicoid protein tells the cells along the axis where they are and what structures to form.
And they proved this.
They did. They could inject bicoid mRNA into different parts of an embryo and induce head structures there. It was definitive proof of a specific morphogen establishing a body axis, highlighted the crucial role of the mother's genes, and cemented the gradient concept as fundamental to development.
Okay, this intricate gene control raises a really old question. When a cell differentiates, like becoming a skin cell, does it throw away the genes it doesn't need anymore? Or does it keep the whole instruction book?
That was a huge debate for a long time, but we now know, for the most part, differentiated cells retain their full genetic potential. They haven't lost genes, they've just silenced the ones they don't need.
And how did we figure that out? Organismal cloning seems key here.
Right, cloning making a genetically identical copy of an organism from one of its cells without sex. The early breakthroughs were actually in plants. Back in the 50s, F.C. Stewart showed you could take a single cell from a carrot root, put it in culture, and grow a whole new identical carrot plant.
So that carrot root cell was two-tippedent. It had the total potential to become any part of the plant.
Exactly. Plant cells are remarkably flexible. Animal cells proved much tougher. Differentiated animal cells usually don't like to divide in culture or revert back.
So they used nuclear transplantation, taking the nucleus out of a differentiated cell and putting it into an egg cell whose own nucleus was removed.
Yes, pioneered by Briggs and King, and then significantly advanced by John Gurdon with frogs. He showed that if you took a nucleus from an early frog embryo cell, it could often direct the development of a normal tadpole.
But if the nucleus came from a more differentiated cell, like from a tadpole's intestine,
The success rate dropped way down. The older, more specialized the donor nucleus, the less likely it was to work properly. This suggested that while the genes weren't lost, something was happening to the nucleus during differentiation that made it harder to reset. Gurdon's work earned him a Nobel Prize.
and then the big one, Dolly the sheep in 1997, cloned from an adult cell.
A huge moment. They used a cell from the mammary gland of an adult sheep. It proved definitively that the nucleus of a differentiated mammal cell could, under the right conditions, be reprogrammed to direct the development of a whole new animal.
But it wasn't exactly smooth sailing, was it? Dolly had issues, other clones had problems.
Definitely not smooth. The success rate for cloning mammals is still very low. Cloned animals often suffer from various health problems. Dolly had arthritis early. Cloned mice often get obese, have organ issues, die prematurely. Even clones that look normal might have underlying problems.
And they aren't always perfect copies either, like Cece the cat.
Right. Cece, the first cloned cat, had a different coat pattern than her mother or nucleus donor. That's because coat color patterns in cats like that are partly determined by random X chromosome inactivation during development. It highlights that even with identical genes, random events and environment play a role. Identical twins aren't perfectly identical either.
So why is cloning so difficult and prone to errors? It comes down to epigenetics, doesn't it?
Exactly. Differentiated cells have specific patterns of epigenetic marks, things like DNA methylation or histone modifications that silence certain genes. These marks are stable and help maintain the cell's identity.
But for cloning to work, you need to erase all those adult epigenetic marks and reset the nucleus back to an embryonic state.
And that reprogramming process during nuclear transfer is often incomplete or faulty. The DNA in cloned embryos frequently has incorrect methylation patterns, which messes up gene expression during development and leads to the abnormalities we see. It really shows how crucial that epigenetic layer of control is.
Okay, so reprogramming adult cells is tough. What about cells that are naturally flexible? Stem cells.
Right, stem cells are defined by two key properties. They are unspecialized, and they can divide indefinitely, producing more stem cells, and they can differentiate into specialized cell types under certain conditions.
There are different kinds, right? Embryonic stem, ES cells.
Are harvested from the inner cell mass of a very early embryo, the blastocyst. These cells are pluripotent, which is a really important term. It means they can differentiate into any cell type in the adult body. Plus, they can be grown indefinitely in the lab.
And then there are adult stem cells.
Yes, found in various tissues in the body, bone marrow, brain, skin, etc. Their job is to replace cells that wear out or are damaged. But they're generally not pluripotent. Their potential is more limited. They can usually only make a few specific types of cells relevant to their tissue of origin.
The excitement around stem cells, especially ES cells, was huge for regenerative medicine. The idea of growing replacement tissues.
Immense potential. Imagine growing new insulin-producing cells for diabetics, or neurons for Parkinson's patients, or heart muscle cells after a heart attack. Therapeutic cloning was the idea of making patient-specific ES cells to avoid immune rejection, but it came with significant ethical debates because it involved creating and destroying embryos.
But then came a massive game changer, induced pluripotent stem iPS cells.
Absolutely revolutionary. In 2007, Shinya Yamanaka figured out how to take fully differentiated adult cells, like skin cells, and reprogram them back into a pluripotent state, making cells that behave very much like embryonic stem cells.
Just by adding a few specific genes.
Essentially, yes. Using viruses to deliver just 4 key stem cell master regulatory genes, he could reset the epigenetic landscape and induce pluripotency. Yamanaka shared the Nobel Prize with Gurdon for this work.
And the advantages are huge. No embryos involved, potentially.
Right. It bypasses many of the ethical concerns surrounding ES cells, and the potential uses are incredible. You can make iPS cells from patients with genetic diseases to study the disease mechanism in a dish, like creating neurons from a Parkinson's patient.
And for therapy.
You could take a patient's own skin cells, turn them into iPS cells, differentiate them into the needed cell type, say retinal cells, and transplant them back into the patient. Because they're the patient's own cells genetically, there's no risk of immune rejection. This is already happening in clinical trials, like Dr. Takahashi's work in Japan for macular degeneration. It's personalized regenerative medicine becoming a reality.
And now they're even looking at direct reprogramming, skipping the iPS stage.
Yes, trying to convert, say, a skin cell directly into a neuron or a heart cell. It's an even more advanced frontier, trying to make the process more efficient and direct. The whole field is moving incredibly fast.
Okay, so we've seen the beauty and complexity of development and repair. Now let's pivot to when things go wrong. When that precise gene regulation breaks down, leading to cancer.
Cancer is essentially a disease of uncontrolled cell growth and division. Cells escape the normal rules, and the underlying cause is almost always genetic changes, mutations in somatic cells. These can be spontaneous errors during DNA replication, or caused by carcinogens, radiation, some viruses.
And specific types of genes are usually involved. Proto-oncogenes.
These are normal, healthy genes. Their job is to produce proteins that stimulate cell growth and division when needed. Think of them as the gas pedal for the cell cycle.
But they can mutate into oncogenes.
Right. An oncogene is a cancer-causing gene derived from a proto-oncogene. The mutation makes the gas pedal stuck in the on position. This can happen if the gene gets moved to a highly active region of the chromosome, or if the cell makes too many copies of the gene, amplification, or if a point mutation makes the protein product hyperactive or resistant to breakdown. The result is excessive cell division.
So that's the accelerator. What about the brakes?
Those are the tumor suppressor genes. Their normal job is to inhibit cell division to prevent uncontrolled growth. They act like the brakes on the cell cycle.
So if they get mutated,
If a mutation inactivates a tumor suppressor gene, the brakes fail. The cell loses a critical mechanism for stopping proliferation when it shouldn't happen. This also contributes significantly to cancer development.
what kinds of jobs do these tumor suppressor proteins do?
All sorts of crucial things. Some repair damaged DNA, preventing the accumulation of more mutations. Some control cell adhesion, helping keep cells anchored properly, which cancer cells often lose. Others are involved in signaling pathways that can halt the cell cycle if something is wrong.
The really famous one is P53, the guardian angel of the genome.
That's the one. P53 is activated by DNA damage or other cellular stress. It can then switch on genes that halt the cell cycle, giving the cell time to repair the DNA. If the damage is too severe, P53 can trigger apoptosis programmed cell death, telling the damaged cell to sacrifice itself for the good of the organism.
So losing P53 is really bad news.
Extremely bad. If P53 is mutated or missing, which happens in more than half of all human cancer, cells with damaged DNA can just keep dividing, accumulating more mutations, and heading down the path to cancer much faster. It's fascinating. Actually, elephants have many copies of the P53 gene, something like 20 copies compared to R1, and they have remarkably low cancer rates despite their size and lifespan.
That makes sense. So cancer isn't usually just one bad mutation then. It's a series of unfortunate events.
Exactly. That's the multi-step model of cancer development. It generally takes multiple mutations, accumulating over time, maybe half a dozen or so key hits, to turn a normal cell into a fully malignant cancer cell. This is why cancer risk increases significantly with age. There's simply more time for these mutations to pile up.
Colorectal cancer is often used as an example of this stepwise progression.
Yes, it's a well-studied model. It often starts with a benign polyp, which might have lost a key tumor suppressor gene, like APC. Then maybe it acquires an activating mutation in an oncogene, like RAS. Later, perhaps it loses P53. Each hit gives the cells a growth advantage, leading progressively towards malignancy. That's why screening like colonoscopies is so important. You can catch and remove polyps before they become fully cancerous.
And inheriting one faulty gene, like a mutated BRCA1 or BRCA2, for breast cancer risk.
That doesn't guarantee cancer, but it means every cell in your body already starts with one hit. You're one step closer to accumulating the necessary multiple mutations, which significantly increases your lifetime risk.
Finally, how is all this molecular knowledge changing how we treat cancer?
It's transforming it. We're moving towards personalized cancer treatment. Advances in DNA sequencing allow us to analyze the specific mutations in a patient's tumor. We now understand that breast cancer, for example, isn't one disease. It's multiple subtypes defined by which genes are active, like receptors for estrogen, ERI, progesterone, PR, or HER2.
And knowing the subtype means you can choose the right drug.
Precisely. If a tumor is ER positive, drugs like tamoxifen that block estrogen signaling can be very effective. If it overexpresses HER2, drugs like Herceptin that target the HER2 protein can work wonders. Other types might respond best to specific chemotherapies. By tailoring the treatment to the tumor's specific molecular profile, we can significantly improve outcomes and reduce side effects. It's a much smarter, more targeted approach.
It really has been an incredible journey, from that single cell to the complexity of life and understanding what happens when the controls go wrong. We've covered the precision of development, differentiation, apoptosis, pattern formation.
The amazing potential and challenges of cloning and stem cells and the intricate molecular pathways involved in cancer, it all comes back to gene regulation, doesn't it? Controlling which genes are on or off at the right time and place.
So what's the big takeaway? Why does all this matter?
Well, it's about understanding ourselves, how life is built, how it maintains itself. And critically, it gives us the knowledge to potentially repair damage, regenerate tissues, and fight diseases like cancer far more effectively by targeting the root causes. It's fundamental biology with profound real-world impact.
Absolutely. And it leaves you with a final thought, perhaps. Given the incredible, almost unbelievable precision needed for development and all ways gene regulation can go wrong,
Yeah.
what does that really tell us about how resilient life is, but also maybe how fragile it could be too?
That's a deep one, something to definitely ponder.
Thank you for joining us on this deep dive.